Christmas is almost here, and while everyone is running around, purchasing, packaging, and sending Christmas presents to one another, our cells are also hard at work, synthesizing, packaging, and sending essential integral membrane proteins to their various organelles. Come with me as we take a closer look at the cell's fascinating protein delivery process, the endomembrane system. Each integral membrane protein begins its journey as it is synthesized in the rough endoplasmic reticulum near the nuclear membrane. From there, proteins are grouped together in small vesicles, which travel from the endoplasmic reticulum, or ER, to the endoplasmic reticulum Golgi intermediate compartment, or ERGIC. From there, they proceed in new vesicles to the multi-layered Golgi complex, which serves as a massive intracellular FedEx center. Here, proteins are modified, grouped together by destination, beautifully packaged into coated vesicles, and shipped out to any of several membrane locations via cytoskeletal microtubule highways. Some of these proteins are destined for display on the exterior of the plasma membrane. Others will be delivered to destructive lysosomal organelles. And, as happens every Christmas season, some of these packages will be returned to the original ER for a refund. Now that we have a general idea of a protein's movement through the endomembrane system, let's take a closer look at the shipment processes, and especially the coat protein wrapping paper of these endomembrane vesicles. The endoplasmic reticulum, Ergic, and Golgi each have unique functions regarding the translation of proteins and assembly of vesicles. The translation of nascent proteins from free-floating ribosomes around the rough ER pauses while the ribosome embeds in the membrane. Next, proteins complete translation and are properly folded within the endoplasmic reticulum exit sites, or ERES. COP2 vesicles arise from the assembly of COP2 coat protein complexes forming around these sites, a process which we'll discuss later in greater detail. These vesicles move forward through the system from the ER to the ergic. The ergic is formed by homolytic fusion of these vesicles. The first major level of protein sorting occurs in the ergic. Escaped ER resident proteins are gathered and returned back to the ER through specialized COP1 coded vesicles. These proteins are recognized by a KDEL, lysine, aspartic acid, glutamic acid, leucine, amino acid sequence on their carboxy terminal end. This binds to a KDEL receptor in the ergic membrane. These receptors have a KKXX sequence, which is recognized during the formation of COP1 vesicles. These vesicles then return to the ER in a process called retrograde vesicular transport. Proteins destined for membranes other than the ER are further transported to the Golgi apparatus in a process called anterograde vesicular transport. Vesicles arrive at the cis side of the Golgi apparatus and move through to the layered cisternae to the trans side. Each of the cisternae contain particular concentrations of enzymes. These enzymes perform modifying glycosylations to these proteins. Glycosylation is the process of controlled addition of sugars to an organic molecule or protein. There are two types of protein glycosylation, N-linked and O-linked. In the ER, oligosaccharides assembled from dialcohol phosphate foundations are transferred to nascent protein asparaginins. This is called N-linked glycosylation. O-linked glycosylation structures are built from the amino acids serine and threonine. This modification takes place in the Golgi and serves as a label to help with vesicle cargo specificity, immune system recognition, control of cell metabolism, and more. Next, we'll take a look at the formation, shape, and function of vesicle coat proteins. Coat proteins serve two main functions: selective capture of cargo proteins within the donor compartment, and the formation of membrane curvature resulting in various transport vesicles. There are also two major types of vesicle coat proteins, COP1 and COP2. Let's examine the differences in the formation of these coats and the factors that affect their shape. COP1 coated vesicles form through the action of ARF1, a small GTPase, and a heptameric protein complex called codamer. The codomer can be broken down into two subcomplexes: a trimeric assembly of Ret1, Sec27, and Sec28, and a second complex composed of Sec26, Sec21, Ret2, and Ret3. The polymerization of these 7-protein COP1 complexes results in the encasement of retrograde vesicles. To help us illustrate the formation of COP2-coated vesicles, I've enlisted the help of my handsome, protein-infused friend, Charles. COP2 coat assembly begins at the ER membrane. A SAR1 protein, represented by Charles, becomes embedded in the exterior leaflet of the membrane. Upon arrival, the SAR1 protein is bound to GDP, but as it becomes membrane associated, a guanine nucleotide exchange factor, or GEF, phosphorylates the GDP to GTP. This brings the SAR1 great joy. Then, the SAR1 GTP complex recruits a heterodimer of two proteins, Sec23 and Sec24. Sec24 selects cargo proteins by directly binding to ER export signals. Next, heterotetramers of Sec13 and Sec31 proteins are recruited through interactions between SAR1, Sec23, and Sec31. Several of the SAR1 complexes will link together to form a spherical cage-like structure which encases the vesicle. These coat complexes can lead to the development of three vesicle shapes: narrow cylindrical tubes, broad flattened cisternae, and spheres of varying sizes. When COP2 coat proteins pinch off vesicles from the membrane, they generate a curved shape due to flexion of Sec13 and Sec31 rods, and an inner coat layer composed of packed lattice on tubular liposome vesicles. The flexion allows for the spherical shape while leaving the potential for other shapes if needed. A special protein, Sec16, is also involved in delaying scission of nascent vesicles, allowing them to grow to the required size. The spherical or tubular budding of COP1 vesicles is determined by the lipid modifying enzyme lysophosphatidic acid acyltransferase gamma and phospholipase A2 alpha. Enhanced scission of carriers by lysophosphatidic acid acyltransferase gamma results in spherical COP1 vesicle formation, while inhibited fission by the phospholipase A2 alpha enzyme leads to vesicular tubulation. As you can see, the intracellular protein production and delivery system includes an intricate and extensive network of precise and necessary components. Without any one of these proteins, organelles, cytoskeletal components, chemical signals, or other component, the cells could not possibly produce and deliver all the integral proteins required for multicellular life. This incredible integration and cooperation of the endomembrane system serves as a beautiful testament to God's incredible design. As we continue to study the amazing inner working of the cells and organisms in creation, we must be careful not to lose sight of the Creator. King David summarized an appropriate posture of adoration in Psalm 139 when he sang, "For you, O God, created my inmost being. You knit me together in my mother's womb. I praise you because I am fearfully and wonderfully made. Your works are wonderful. I know that fully well." Psalm 139, 13, and 14.